All right, hello Bio30s. We're gonna try this again for our second lesson on genetics. Hopefully you guys did the non-Mendelian questions on page 18 and 19 of your note package. Non-Mendelian stuff like incomplete dominance, codominance, epistasis, polygenetic inheritance. So there's quite a bit of stuff there. Make sure that you do take a look at those questions. I'm still unsure of how we're going to be doing assessments. I don't know if I'll be giving you online quizzes or exams, or if you'll come into the school in small numbers. I have no idea. Nobody really knows. We're all kind of in this together. We're doing our best and we're trying to figure it out. I still haven't heard anything about diplomas. I'm really hoping that they cancel them, but as of recording right now, I'm recording this before spring break. I don't know. I don't know what's going on, but as of right now, it's on. So we'll make the best of it, and we'll continue on with our genetics. Okay. So today we're gonna do another lesson on sex-linked inheritance. So it's another type of non-Mendelian genetics. But before I do that, I would like to go through one question from your homework. If you haven't done your homework yet, please go do that. Page 18 and 19 in your note package. And again, all the answers are on the Google site. But I will pick one question that gave my classes in the past a bit of a tough time. Okay, so if you haven't done the homework, please go do that now. Pause the recording and come back after you've done it. Okay, this one has given my classes a bit of a problem in the past. And it's because of these numbers up here. So in real life, when we do these experiments, sometimes the ratios don't work out perfectly, right? Like we talked about a 9 to 3 to 3 to 1 ratio in Mendelian genetics. We talked about 3 to 1 ratios, and you have a 25% chance of doing this and a 75% chance of doing that. But in real life, we don't get perfect ratios, right? So here, we have 105 plants that are round, 199 plants that are oval, and 97 that are long. So if you were to actually do this out and divide by number, you would not get a nice perfect whole number ratio. Okay, but let's take a look at what's going on. So in this cross, we have two types of radishes. Okay, we have round radishes and we have long radishes. And when we put these two together, we get a third type of radish. We get an oval radish. Okay. Now, this isn't a plant with round radishes and long radishes, right? This is a third phenotype. So this would not be codominance. This would be an example of incomplete dominance. Okay. If this was codominance, we would get both round and long radishes in our cross. But because we get something that's completely different from the parents, round plus long equals oval, this tells us that this is incomplete dominance. Okay, so what they're doing here, and let's do this. Okay, now again, this is non-Mendelian, so I can't just use big R and little r. I mean, you could, but there's no dominance and there's no recessives here, right? So what we're going to do is we are going to say, okay, so there's shape, S. So there's a round shape. Okay, so this would be round. By the way, I went and bought this little tablet and pen thing, so hopefully it works a little better than in my last presentation. Okay, and then we have shape. That's the trait, and we have long. Okay, now because there's no dominant or recessive here, I'm not going to use big and little because there's no dominant recessive, right? So what I'm going to do is I'm going to just use S superscript R and S superscript L. Okay, so now how would we get a round parent? Okay, now remember, all individuals have two alleles, right? You get one from each parent in the beginning. So how would I write a round parent? Well, a round parent would have two round alleles. Okay, that makes them round. And how do I get a long parent? Well, I would get a long parent that would look like this. Okay, so I couldn't have any other way, because if I had an R and an L, knowing that R and L are codominant, that's what gives me my oval. Okay, so if this was my parental, this is my parental generation here. Okay, I take a round and a long and cross them together. Okay, and what I get from this is I get one parent giving the round allele, and I get one parent giving the long allele. So again, if you wanted to do a Punnett square, you could do a Punnett square of this cross. I'm hoping by now, though, you guys kind of understand this, right? So I am skipping steps, but this is what we would do, right? And 100% of my F1 generation would be round and long, and this would equal oval. Okay, so that's oval. Now, they want to say here, what's the approximate ratio of the F2 generation? So in order to get F2 generation, what I need to do is I need to take two of my F1s, so SR, SL, that's oval, okay, times another oval. So I'm crossing two oval plants from my F1. And when I do my Punnett square, okay, so SR, one parent can make two different types of gametes, the other parent can make two different types of gametes. What I end up with here is I get an SR, SR, I get an SR, SL, get an SR, SL, and I get an SL, SL. So my approximate ratio in my F2 would be round, okay, to oval to long. Okay, that's my three phenotypes. I have one round to two oval to one long, right? One round, two oval, one long. So my ratio is a 1 to 2 to 1 ratio. Now that's probability. That's just in a perfect world, right? It's like rolling a dice. What's the probability of getting a 1 on a 6-sided dice? Well, it's 1 out of 6. Does that mean if you roll the dice 6 times, you're definitely going to get a 1? No, you could get 6 sixes, right? You could get 2, 3, 4, 5, 6, and a 5 again, right? So these are just theoretical probabilities. Now in real life, what we get is we get this 105, 199, 97 ratio up here, right? So they did the cross in real life, and this is what they got. Well, if we round this, okay, and again, this is biology, right? So we're not too concerned about, being perfect with our math, but if we rounded all of these, we get a 100 to 200 to 100, which when we reduce it is a 1 to 2 to 1 ratio, kind of, almost, right? We're close. That's close enough. So do the ratios match up? Yes. This is what we would expect. Okay, is it perfect? No, it's not perfect 1 to 2 to 1, but when we round it, we see that it's darn close. Okay, so yeah, that's how you would do this question. And again, a lot of people kind of get confused because these numbers are not a perfect 1 to 2 to 1 ratio, but they are close. And in real life, as we know, probabilities, theoretical probabilities, and actual events are sometimes different. Okay. What we're going to talk about is sex-linked inheritance. So this is another type of inheritance pattern that Mendel didn't see. Okay, so you have to understand Mendel was around the late 1800s when he was doing stuff. He did not know about mitosis. He did not know about meiosis. He didn't know what gametes were. Well, he knew there were gametes, but he didn't know what was inside. I remember, our discussion of the homunculus, right? Van Leeuwenhoek, when he looked in his microscope, he didn't know what was inside of a sperm. He thought it was a little blank person. So, a lot of these guys were just kind of making educated guesses, right? Um, and it turns out that for certain traits, Mendel was bang on. Okay, but he did kind of open the door. Now, in the early 1900s, there was a guy by the name of Sutton, and Sutton was doing was doing research. And what he was noticing is that there were these things inside the nucleus of cells that appear during cell division. Okay, so he called them chromosomes because they were colorful when you stain them. They picked up stain, and chromo means color. So basically, there were these structures, and again, he didn't really know what they were. He didn't know they were made of DNA or anything like that, but he kind of thought that there might be genes located on these chromosomes. Okay, so they were looking at what's going on. Like they were taking Mendel's research and saying, okay, yeah, Mendel was right in a lot of areas, but why? What was going on there? So Sutton started looking at these chromosomes, and he noticed that in males and females, there were some chromosomes that were different, right? So in females, they had all the chromosomes were you could put them into pairs. In males, there was one pair that didn't quite line up, and we now know that as the sex determining pairs. Okay. Now, at the same time in California, there was a guy by the name of Thomas Hunt Morgan, and Thomas Morgan was trying to replicate Mendel's experiments, but instead of using pea plants, he was using fruit flies. Okay, now these fruit flies are the same little annoying flies that if you leave bananas on the counter, um, they all of a sudden appear, right? These guys are harmless little things, but they're pretty simple. They have pretty simple chromosome arrangements, and they have some traits that are either like Mendel's pea plants that are discrete. We call discrete. So, for example, they either have red eyes or white eyes. Okay, they either have wings that are very long or they have wings that are very short and don't work. So they have lots of traits that have an either or type of condition. Okay, so he was using these fruit flies, and they're really easy to grow and they're really easy to reproduce, and they don't take nearly as much time or space as pea plants. Okay, so he was trying to replicate Mendel's experiment. Now, in one of his first experiments, what he did is he took a fly that had a mutation that had white eyes, okay, and he knew that this was a recessive condition, okay. So white eyes, he said, was little r, okay, and then he took some flies that had red eyes, and he knew that this was the big R, okay. Now he took a male with white eyes and he crossed this male with a female that had red eyes. Okay, and what he found was that all the F1 population had red eyes. Okay, and this told him that this was the dominant condition. Okay, so again, right, if we took red, big R, pure breeding, right, times white, little r, we would expect to get 100% of my F1 generation to be red eyed, right? So again, if this was Mendelian, big R, that's my red eyed female, times little r, it's my white eyed male. 100% of my F1 generation should be big R, little r, which would be red, right? And that's what he found. So next step, just like Mendel, he's going to take two members of his F1 generation and he's going to cross them together. And what should he have got? Well, if this was a true Mendelian ratio, he should have got a 3-to-1 ratio in his F2. Okay. So what Morgan found is that in his F2 generation, he found that all the female flies had red eyes. Okay. So again, before we never looked at the difference between males and females, right? We never said, oh, look, females are different than males. It was just, if we were doing blood type or if we were doing coat color in rabbits or whatever, we never said that, oh, the females are different somehow from the males. But here all of a sudden, in his F2 generation, it should have been 3-to-1 ratio red to white in his females, and it should have been 3-to-1 ratio in his F2 males, right? There should have been no difference between males and females, but all of a sudden he found that there was. Okay, he found that all the female flies in the F2 generation had red eyes. It wasn't 3-to-1 like he thought it would be. And he found that there was a 1-to-1 ratio in his males. So half the males had red eyes and half the males had white eyes. So again, not a 3-to-1 ratio. Okay, so what happened? What happened to the 3-to-1 ratio? And why were the sex of the individuals, why did it come into play? Okay, what happened there? Well, it turns out, and again, this is where Sutton comes in because Sutton was dealing with chromosomes, right? So Sutton, in his lab, when he's dealing with chromosomes, he found that, like in humans, fruit flies have two X chromosomes in the females, and in the males, they have an X and a Y. Okay, so they have a sex determining pair. And what we haven't taken into account up to this point is that there are some genes for some things that are on the X and Y chromosomes. So, of course, if they're on the X chromosome, then it would be different for females, and if it was on the Y chromosome, it would be different for males. So, everything that we've talked about up to this point was on the other chromosomes, the homologous chromosomes. Remember what those are called? You guys remember what the other 22 pairs of chromosomes were called? They were called autosomes. Okay, so everything up to this point we've been talking about were on the autosomes, so it didn't matter if you were male or female. Now all of a sudden, eye color in fruit flies is not carried on the autosomes, it is carried on the sex chromosomes. So things are inherited a little bit differently. Okay, so when Thomas Morgan, he sat down and he said, okay, what's going on here? Why am I not getting these Mendelian ratios? What he found was that when he assigned the alleles to the X chromosome, he got some things happening a little bit differently. Okay, so keep in mind that we do have red is dominant. Okay, and we do have white as recessive. So we do have a dominant recessive relationship here. But these alleles are carried on the X chromosome. Okay, so when we had our white-eyed male, okay, here's the white-eyed male, he only had one allele because the Y chromosome did not carry the other allele because it's not homologous, right? The X and Y chromosomes are not homologous in males, meaning they're not the same size, the same shape, and they don't carry the same genes at the same location. Now, the pure breeding red-eyed female had two X chromosomes, and she carried a big R on each of those chromosomes. Okay, so when we're making gametes, when a male makes gametes, it's the male that determines the sex of the offspring, by the way. And the reason why is because the male carries an X chromosome and the male carries a Y chromosome. So those are the two types of sperm the male would make. Now, this pure breeding female, she can only make one type of eggs, and those are eggs with R in them. Now, why am I using X and Y? Because this is a sex-linked trait. Okay, that means that the trait is linked to the sex chromosome. So when this egg and this sperm get together, okay, we get a red-eyed female. When this egg and this sperm get together, we get a red-eyed female. Okay, when this egg and this sperm get together, we get a red-eyed male. And when this egg and this sperm get together, we get a red-eyed male. Okay, now when we take two members, this is the F1 generation. When we take two members of the F1 generation and we cross them together, why don't we get a 3-to-1 ratio? Well, because males only get one X chromosome. Their other chromosome is a Y chromosome, right? So let's see what our F2 looks like. So this would be my F1 generation. These guys over here are going to be my F2 generation. Now again, if this was Mendelian, we'd expect just a 3-to-1 ratio, right? But we got different ratios of males and females. Okay, so there's my F1 female, and there's my F1 male, red eye. Both of them are red-eyed. Okay, so here we have red-eyed female, red-eyed female, right? 100% of the females are red-eyed. Here we have a red-eyed male, okay, and now we have a white-eyed male. Okay, so 50% of the males have red eyes and 50% of the males have white eyes. So This is not a 3-to-1 ratio because on this side we have the females and they're all red. On this side we have the males, half of them are red, half of them are white. So this is not a 3-to-1 ratio. Okay, so this does not follow regular Mendelian genetics. And the reason for that is because Mendel did not take into account this idea of chromosomes and especially that males and females would have different. Okay? There are certain traits that are carried on the X chromosome, certain traits that are carried on the Y chromosome. We have to be careful of this. Okay, so here is a question. This is an old diploma question. And this deals with hemophilia. So hemophilia is a disease where you lack a certain protein when you're clotting your blood. Okay, so if you were to cut yourself or if you were to bump yourself and cause bleeding internally, like a bruise or something, those of us who do not have hemophilia, we have all the proteins that are involved in that clotting process that you learned in Bio 20, right, with the platelets and the thromboplastin and all that kind of stuff. Okay. Now, people with hemophilia, they lack those proteins, so their blood doesn't clot and they can bleed to death. Okay. So hemophilia is a very serious disease. Now, what we find is that men get hemophilia more often than women. Okay. And that has to do with the fact that it's carried on the X chromosome. Now, remember, females get two X chromosomes, so they always have a backup, right? And this is a recessive disease. So if you only have one bad gene and you have a good gene, the good gene will if it's dominant, it'll kind of overcome the bad gene. But if a male gets a bad gene on its X chromosome, he doesn't get another try, right? He gets a Y chromosome and it's not carried there. So when do we know when to use X and Y? When the question says this. Okay, if the question does not say the trait is X-linked or sex-linked, sometimes you'll see sex-linked. Okay, if the question does not say this, then we do not use X and Y. We just use our regular notation because if it's on an autosomal chromosome, it doesn't matter if you're male or female because we all, our 22 pairs of chromosomes are the same in males and females. It has to say in the question the trait is X-linked or sex-linked. Okay. Now, many students after this lecture, they use X and Y for everything. Anytime it says, oh, we have a male cow that's red, or a male bull that's red and a white cow, and they cross, and all of a sudden they're using X, X, and X, Y for everything. No, only if it says this. If it doesn't say this or this in a question, just use regular old genetics. Okay, but this question does say it's the hemophilia is X-linked. Okay, so H, big H, normal. Okay, little h, hemophilia. All right, so when we are doing this question, because it says the trait is X-linked, we have to attach these alleles to a chromosome. Okay, so it tells us that a female is hemophilic. So that means she has the disease. All right. So because it's X-linked, we have to denote that she's female using two X's. Okay. Now, what's the only way to be a hemophiliac because it's recessive? Well, in a female, it means that both of your X chromosomes have to carry the faulty gene. Okay. So she would be homozygous recessive. Okay. Because she carries two X chromosomes and both X chromosomes have the faulty allele. Okay, now she marries a man who is not a hemophiliac. Okay, which means that his X chromosome, and males only get one X chromosome, has a good gene. It knows how to make the protein for blood clotting. And then he's got a Y chromosome. And do I put an H here? The answer is no. Okay, H is not carried. The gene for blood clotting is not carried on the Y chromosome. These are not homologous. So I don't have that gene here. Okay. What is the probability of this couple having a child that is hemophilic and the sex that that child would be? Okay. So if we do a Punnett square for this cross, okay. So she can only give an X chromosome that's faulty. Okay? He can give a good X chromosome, but his other sperm are going to have Y chromosomes in them. Okay? So if these two X chromosomes get together, okay, they have a daughter. And is the daughter going to have hemophilia? The answer is no, but she's a carrier for it, but she got a good X chromosome from her dad, and that chromosome has the gene to make the protein. So she's okay. She got a backup chromosome, right? What about if we put these two together? Again, that results in a daughter, and the daughter's okay, right? She gets a big X from her dad, and she gets a little X she gets a little H from her mom on the X. She gets a big H from her dad on the X, but that's okay. She's heterozygous. She's got a good gene. Now, what about the sons? Sons get their X chromosome from their moms, okay? And they get their Y chromosome from their dads. So that son inherited a faulty X chromosome from mom, and he doesn't get a backup X chromosome. So that's the only instruction he has. So is he going to have hemophilia? Yes. Okay. What about the last combination? Again, because mom only has faulty X's to give, every son inherits their X from their mom, meaning that they're always going to get this bad H, right? So what is the probability of this couple having a child that is hemophilic? 50%. Okay, there's a 50% chance of having a boy, 50% chance of having a girl. And the sex of that child would be male. Okay, now 100% of the males will have it. But what's the chance of them having a child with the disease? 50%. Okay, what will the sex of that child always be that's hemophilic? Male. Okay, here's another one. In humans, muscular dystrophy is a disease that's characterized by the gradual wasting away of muscles. It begins in childhood and is usually fatal by early teens. Okay? This trait is controlled by a sex-linked allele, recessive sex-linked allele. Okay? A couple have an affected son. Give the genotypes of these parents. What is the probability of them having another affected child? Okay, so here we have a bit of a problem, right? It says give the genotypes of these parents. Now, they have an affected son, and this is sex-linked recessive, meaning that it's carried on the X chromosome. Okay, so if mom was X, let's call big N normal, okay, and let's call little n muscular dystrophy, okay, MD. So if mom was X little n, okay, which would work in this question, but here's the problem. Would this woman that looked like this, that had the disease, would she ever have children? Well, it says right in the question that it's fatal by early teens, right? So we would never have a mom that could give birth. I mean, this is a terrible disease, right? You waste away, you're in a wheelchair, you lose control of your body. So there's no way that a woman with muscular dystrophy is ever going to have a child. Okay. So if that's the case, why does the disease exist? Well, what if mom looked like this? Okay. So this would make mom normal, but she's a carrier for the disease, right? Again, same thing with dad. Dad's not going to have muscular dystrophy because he won't be able to have children. All right. So dad is going to be normal. He's going to look normal, right? And be normal. Because he only carries one allele. Now, could these two parents actually have a child with muscular dystrophy? They both look normal, right? They're not dying of muscular dystrophy, but could they have a child with the disease? So mom can make two types of gametes, okay? Dad can make two types of gametes, okay? So if these two get together, we get a daughter completely normal, right? Oops, there's a little. If these two get together, we get a daughter who is a carrier for the disease. If these two get together, we get a son who's normal. And if these two get together down here, we get a son with the disease. Okay. So a couple have an affected son. Is that possible if they're both look normal? Yeah, absolutely, because mom's a carrier. Give the genotypes of these parents. Well, again, could mom technically be X little n X little n? Yes, but she wouldn't be able to have children, right? So, um, in the real world, a mom that's going to pass this gene on to her sons is going to be heterozygous. Okay, so that's the genotype of mom. Dad is normal. Okay, again, he wouldn't father children if he wasn't. So we do our Punnett square. What's the probability of them having another affected child? Again, it doesn't say the probability of them having another affected son. It says child. It would be a 1 in 4 chance. So this condition only happens in boys, never girls. Why is that? Well, in order to happen in girls, we would have to have a dad that has the condition and a mom that has the allele, but a dad that has the condition is not going to father children. Okay, not going to be able to have kids. Since boys with this trait die before puberty, right? So they're not having children. Since they die before puberty, why does this disease exist in the population? Because moms can be completely normal, but they can carry the allele. Okay, and that's why it gets passed down. Like this daughter right here would have no problems, okay, if she was born with that genotype, but she is carrying the allele. So that's why it persists in society. Okay, let's try another one here. You guys try this one. All right, I want you guys to try this one. And so stop the video right now. Give this one a try, and then start the video after you've tried it, and I'll work through it. Okay, so I'll give you a couple seconds here. Just pause the video. Okay, so hopefully you've tried this, and you're not just kind of putting this on in the background, right? You need to actually try these, right? It's practice makes perfect. So if a mother carried the sex-linked gene for color blindness, okay, and father had normal vision, okay, so again, it says right in the question, it's sex-linked. So mom is a carrier. Now, carrier equals heterozygous, okay? This is one of those words in genetics that you need to know. It's like test cross and roan. Carrier means heterozygous. So mom was carrier, okay? So we're going to say that, I don't know, blind. Let's use N, big N for normal vision, okay? And use little n for color blindness. I don't want to use C's because it's too hard to tell the difference, okay? So mom is a carrier for this disease, meaning mom has normal vision, okay, because she gets a normal allele, but she's a carrier. A carrier means that she shows no sign of the disease, but she's carrying the allele. Now, dad has normal vision, okay? So dad has a big X and dad has a Y. Again, it's sex linked, so we need to use X and Y. Okay? What would be the probability of them having a colorblind son? So we need to do our Punnett square. Okay, so mom, X with the big N. Now, I'm putting mom on the top here and dad on the side, and sometimes I put dad on the top and mom on the side. Does it matter where I put them? No. Okay, so dad is over here. He's normal vision, but he's got a Y chromosome. So up here are going to be daughters, right? So normal vision daughter, normal vision daughter. Okay, but she's a carrier. Now what about the sons? Okay, normal vision son, colorblind son. Okay, so what would be the probability of them having a colorblind son? Now again, what's the probability of them having someone that's colorblind? Okay, and it being a son? Well, right here, 25% chance. Okay. All right. Okay. Okay, here's another example problem. So I would like you guys to pause the video here, and I want you guys to give this one a shot. Okay, so pause the video right now, give this one a try, and then once you've tried it, unpause the video, and I will go through it. Okay, so pause it now. Okay, so let's go through this one. This one's a little bit different, and my pen is not behaving. Let's see what's going on here. Bought this fancy pen to see if it would be better than my mouse in the last presentation, and for the most part, it's much better, but it's acting weird. Okay, so genetic disease caused by a dominant allele carried on the X chromosome. So it tells us it's carried on the X chromosome. It's sex-linked, so we have to use XX for female, XY for male. But the difference is a dominant allele. Okay, so in this case, big P is not normal. Big P is actually the disease. Okay, and it's little p that's actually normal in this case. Okay, so the dominant allele actually causes the disease. Okay, so a female with hypophosphatemia, all right, so she is going to be X with a big P, but we don't know what the other X is. Okay, her father had the disease. Okay, so father was X big PY, so that's probably where she got the X chromosome from. And the mom did not have the disease. So mom is going to be X little p, okay? Because she didn't have the disease. So that means that this female is going to get a little p from her mom, okay? So she's X big P, X little p. Now she marries a normal man, and the man is going to be X little p, Y, okay? They will likely transmit the disorder to who? So we do our Punnett square. Okay, so we figured out that mom is going to be X big P. She has the disease, and because her mom didn't have it, meant she got a good X chromosome from her mom. In this case, good means recessive. Okay, and dad, the father here, or the husband, I guess, let's say the husband. Okay, he has an X little p and a Y because he has normal vision. Okay, so this daughter would have it. This daughter would be normal. This son would have it, and this son would be normal. Remember, normal is recessive in this condition. So, who would they most likely transmit the disorder to? Her sons only? No. Her sons and daughters equally. All of her daughters, but none of her sons. All of her daughters and 50% of her sons? No. This couple would give it to their sons and daughters equally. 50% chance that their son would have it, and 50% chance that their daughter would have it. Okay. All right, so please look at the questions on page 22 of your note package. Okay, these are all questions dealing with sex-linked inheritance. Yeah, we're just going to keep going on like this. Again, I don't know, I'd normally be giving you a quiz right about now on all this non-Mendelian stuff. Obviously, that's, might not happen in class. Maybe I'll figure out a way to give it to you remotely. I don't know. But for now, this is kind of our new normal. Stay tuned for the next lesson, which will take care of some more genetics inheritance type problems. Okay, and again, just pretend you're in class, and we'll get through this. Okay, we'll figure it all out. All right, take care and stay tuned for an announcement on the Google Classroom about your next lesson.